Do you have written protocols in your laboratory for performing RFLP in your lab?
Yes, we do.
And are those protocols based on the scientific literature?
Yes, they are.
Are there a number of ways to perform RFLP analyses?
There are variations on the theme. I mean, it's basically the same process, but different laboratories use, for example, different restriction enzymes. That's the enzyme that cuts the DNA at a specific site. And also, different laboratories use different probes to look at different sites of genetic variation in the DNA.
Okay. If you will, can we just compare the system that you have with the system in place at the FBI? Are you familiar with that?
Yes, I am.
And how does your system compare with the FBI system?
It's
I think there's a relevancy problem with sustain.
Can I make it? It relates back to databases, Your Honor.
All right, let me see counsel at the sidebar with the court report. Thank you, counsel. Proceed.
Very briefly, Mr. Sims, what's the compatibility or the similarity of the system in place at DOJ lab versus the FBI and many other labs in this country?
the main point is that we use the same restriction enzyme, which is called HAE, H-A-E, Roman numeral 3, and also that we use many of the same probes. There's a battery of probes that overlap. And then also we have exchanged samples with the FBI to show that our data is compatible with their data.
Okay, and if you would you contrast the system that you use in the FBI and many other labs with Cellmark's, because the jury's heard quite a bit of Cellmark's approach to RFLP.
Yes, the main difference is that they're using a different restriction enzyme for their analysis, and I believe it's HNF1 is their restriction enzyme. And what it basically does is it gives larger DNA restriction fragments, so their stuff tends to be higher up on one of these gels that we look at. And they use I believe two of the probes that they use are the same probes as ours. And then I think there are 3 different ones that they also use. But it's the same fundamental process.
Now, Cellmark has described, Robin Cotton described, a cocktail probe, which is a mixture of their single locus probes. Do you or the FBI use that same sort of cocktail?
We do not, and I do not believe the FBI does.
Okay. Do you have controls built into your RFLP testing to measure how well the test has been performed in any given case?
Yes, we do.
Could you briefly describe the types of controls specifically in the RFLP process that you have and what their function is?
Yes, there are 2 main controls that we run on the RFLP on any particular autorad that you see. One will be what is called a K562 standard. That's a DNA, cell cultured DNA that is run almost, not every lab in the nation, but a lot of laboratories in the nation use that as a standard control. We know where the values should be for the 2 bands or sometimes 1 band that is produced by that particular standard. And so that provides us important information. It tells us that the probing was working correctly, and the sizing information is also checked that way. The second control that I mentioned this a little bit earlier, was that we always run on a an RFLP gel, we always run one of these quality control samples, and that is a sample that's unknown to the analyst. And then the analyst goes through and determines what the band sizes are for that particular sample, and then submits that data for review. So that's blind to the analyst.
Okay, and that's, I think you mentioned the supervisor actually looks at what the true values are?
Right.
And if there's a discrepancy between the two, what happens?
If there's a discrepancy between the two, we've never had one that I know of where we've mixed up anything. We have, I know in my case, I had one particular sample where one band, a very high band, was slightly off because of a gel problem, and then we rejected that entire data and I reran the entire experiment.
Okay. If you would, let's talk about how your PCR process works as opposed to the RFLP process. Just give us a little overview. What sort of markers do you use for PCR in the DOJ lab?
Yes. The markers that we use for PCR, first off, would be the DQ alpha marker, and that's the one that has been in perhaps the most widespread use in forensic science. It's got a fairly long history now of usage. Then
Do you know when that was first used in this country?
The first case that I'm aware of that was used in was Dr. Blake, who pioneered the use of PCR in forensic science, and that would be in 1986, I believe. It's the People versus Pestanikas. I think it's a Pennsylvania case. I may be wrong on the state.
So you have the DQ alpha marker,
Yes.
and Robin Cotton described the type of process that entails. Do you have an additional PCR marker?
Yes. The other PCR marker that we have online is called D1S80.
And can you just briefly compare and contrast the method of testing of DQ alpha with D1S80?
Yes. The DQ alpha involves looking at a particular genetic locus that has variation in the DNA sequence, and we can detect that in this typing procedure that we use. So we're actually looking at a little bit of variation in the sequence to detect that polymorphism or different forms. On the other hand, the D1S80 is what is called a length polymorphism. It's somewhat like the RFLP procedure in that the different people have different lengths of this repeated sequence of DNA, but it's detected in a completely different manner than the DQ alpha is.
The jury's heard a great deal of testimony about substrate controls, which we will discuss in a little while. Do you normally process substrate controls through the RFLP process?
No, unless one saw that there was a substantial amount of DNA on the substrate control that could be subjected to RFLP, one would not. They're really for the PCR.
Could you expand on that just a bit more?
Yes. The importance to me of the substrate controls is that provides not only a way of evaluating the substrate that the stain is on, whether it be a sidewalk or whether it be a garment or that sort of thing, but it also provides a way of evaluating how a sample is treated through the entire extraction and testing process. I mean, beginning with the scene, taking it all the way through the initial laboratory examination, and then following it up with the actual testing and typing procedure. And with PCR, one is always on guard for contamination, and that's one way of testing to see whether or not there is some contamination.
Okay, we'll discuss that in a bit. So the substrate controls, if you have them in a given case, are processed through the PCR process from start to finish.
Yes.
And, but not normally in the RFLP process.
That's correct.
Can you give us any idea of how frequently you do not see substrate controls in cases?
We generally get them. We generally ask for them from our submitting laboratories. For example, the Bureau of Forensic Services field laboratories will submit substrate controls to us. But at times, in my experience with certain types of evidence, one cannot always get a substrate control.
Okay, so they're great if you have them?
Yes.
Are there a variety of ways to extract DNA? I think you mentioned the Kelex extraction process.
Yes, there are a variety of ways to do it. Some of them are just modifications of a theme, but there are different approaches, and people have looked at different ways of doing it.
Which extraction procedure do you use in the DOJ lab?
We use what's called an organic extraction. We actually place the sample into a buffer that contains a soap solution and also an enzyme that chews up protein, and that combination loosens up the DNA and gives you what hopefully will be free DNA, because you have to remember that DNA in the cell has protein wrapped around it, and so you have to free the DNA so that you can work with it, and that's what the extraction process is.
And do you have a separate written protocol that describes the PCR process, the DQ alpha process from start to finish?
Yes, we do.
And yet another separate one for the PCR D1S80 process from start to finish?
Yes, we do.
Are they based on the scientific literature?
Yes, they are.
And are these also commercially produced kits that enable you to do these tests?
Yes, the DQ alpha and the D1S80 are commercially available kits, and we use those.
And are the protocols that you used ones that you've drafted yourselves?
Yes, we look at the protocols that are in existence in the scientific literature, and we make some slight modifications of those.
And do the manufacturers of these products provide something in addition, or something called a user guide?
Yes, they provide a user guide that explains some of the parameters of the kits, how they're used.
Would this be a good time, Judge?
Perfect. All right, ladies and gentlemen, we're going to take a 15-minute recess. Please remember all my admonitions to you, and we'll resume in 15 minutes. And Mr. Sims, you can step down.
Back to the answer that the witness gave on time constraints and as to approach. Do you recall that
Yes.
question and answer? And I didn't want to draw more attention to it that had already been done, but the problem I have here is that what Mr. Harmon has done is the defense made a number of motions in this case, both before Judge Kennedy and yourself, for splits of the evidence, and splits early, before there was extensive processing by laboratories. Those motions were denied. Additional motions were made with respect to the pacing of the testing. By bringing out the questioning in the way that he's done, and continually pushing with objections sustained, this whole issue of access and everything else, he puts us in the position now where we have to go into something that I thought we had all agreed under 352 is really not for the jury, is a big problem for all of us, and that is to explain to the jury that the defense asked for half the samples, the defense on a number of occasions asked for portions of the samples early, that these requests were denied, that the testing process has gone in all the way through the trial, that there's been limited access, all kinds of things like that, which I thought that we were going to keep out of bounds. And by asking that question about the time constraint in that fashion, he begins to open the door to this problem, puts us in a dilemma. And I
What do you
think,
propose?
well, I'd have to give that some thought. My real proposal is probably some kind of an instruction from the court with respect, some general instruction on this issue. But I would, again, urge that they not be allowed to go back into this whole issue of, I mean, how many times can we do it now? You know, skating on the edge. I think enough is enough. And I think that and he really went over the line there by implicating all these issues of time constraints and explaining all that to the jury.
Well, it was one question and answer.
I understand that, and I
Right.
tried not to bring undue attention to it, and I did ask for a sidebar, but this is my concern. And I articulated it at the beginning and asked that before these kinds of things came up, that the prosecutor indicate to us he was getting into this area, and I have this abiding concern.
Mr. Harmon,
Yeah,
any
Your Honor,
necessary response?
well, there are always necessary responses to Mr. Scheck. You know, how much longer do we have to pretend that those requests for splits were sincere when they never took advantage of them when we were done
Mr.
with
Harmon,
it.
that's not the issue.
Well, it is, because if
No,
we're going to
Mr.
instruct them on anything,
No, Mr. Harmon, thank you. That's enough. The only issue there was the issue of timing and what kind of time constraints were there. That's really irrelevant at this point. So let's avoid the topic in the future. All right, let's have the jury.
It's not just that, it's this whole question of reanalysis and retesting and
I
what
understand
we
that,
have
counsel.
access to.
I understand. I've indicated to Mr. Harmon, I think it's irrelevant, the timing issues under it's also an undue consumption of the court's time to get into who got to do what when. And I
Yes,
indicated we're not going into that.
well, but if
All
we're going
right?
to go into this
And I've given
reanalysis
Mr.
issue
Harmon
again.
fair warning now. We're not going into that.
Your Honor, give
Let's
me
have the
fair
jury.
warning about what? He's talking about the
No, I'm
potential for
talking about-
reanalysis.
No, we're not going into the timing of who had access to what when.
We're done with that.
Good.
Yeah.
Let's have the jurors. Stand, please. All right, thank you, ladies and gentlemen. Please be seated. Record should reflect that we've been rejoined by all the members of our jury panel. Mr. Sims is again on the witness stand undergoing direct examination. And counsel, we're going to break today at 4 o'clock. All right, proceed.
Mr. Sims, just back up a point I may not have clarified. When you opened items, let's say that had bindles in them, and there were initials and letters and dates on them, you noted those in your report.
I noted those in my laboratory notes,
Okay.
yes. And some of those are noted also in the actual lab report.
On RFLP, why don't you use a cocktail like Cellmark does?
Well, the cocktail is a useful approach because it gives a very quick answer towards exclusion, for example, and that's a very reasonable thing to do. We just have not adopted that approach. We haven't adopted it in the sense that we haven't evaluated that cocktail fully as to if we were taking all our probes and all the conditions to see how it would look, so we just don't use it.
Okay, now, is your lab also investigating or in the process of implementing other PCR markers other than DQ alpha and D1S80?
Yes, we are.
Sustain. Let's move on.
When your lab performs the PCR process, do you use the requisite positive controls and negative controls?
Yes, we do.
And you also, I believe you said you process the substrate controls in the PCR process as if it was a stain item.
Yes.
And in addition to telling you whether or not there's biological material on the substrate closely associated with the stain, do those substrate controls perform a function, a troubleshooting function when they're typed through the whole PCR process?
Yes, if they come up negative, then that shows that you're not picking up any contamination through the entire process, and that would be from the point of collection all the way through the analysis, the typing.
Okay. And would you classify then, in that context, the substrate controls as a negative control?
Well, I generally think of a negative control as something where we know in advance there's nothing. On a substrate control, you don't specifically know that. But I think for our purposes, you can think of that as a negative control, but it doesn't always come up negative because some substrates have material on them.
Okay. Let's talk about how the Department of Justice reviews all the data in a case like this, a complex case where many samples are typed, and what sort of process is involved in reviewing all the data and writing the report. How many people were actually involved, hands-on, in the actual work in this case?
Of the casework analysts, there were 3 of us.
And who are they?
That would be myself, Renee Montgomery, and Steve Myers.
And Renee Montgomery, does she have some specific expertise in one of these areas?
Yes, she does. She is the person who did almost all the D1S80 work in this case.
In this case. And Mr. Myers, what role did he play in this case?
Well, Mr. Myers has special expertise in the extraction and evaluation of DNA from hair samples. So he performed that analysis. He also did some DQ alpha and D1S80 typing.
And who actually then participated in the report writing process?
That would be the 3 of us. I was the main author. Then I reviewed it and got suggestions and input from Montgomery and Myers. And then finally, that entire package is reviewed by the supervising criminalist, Ken Konzak.
And when you say you reviewed things, did you review things such as the DQ alpha typing strips
Yes. In other words, we look at the overall results.
jointly?
Yes.
We're going to get to some autorads in just a moment, but I want to ask you a couple of categorical questions about positive controls throughout the entire PCR process. Did the positive controls type the way they should have through all the samples that were tested in this case?
Yes, I believe they did.
And similarly with the negative controls,
Yes, that would be the extraction blanks and also, which is an extraction reagent blank, and also the negative amplification controls. Those were all negative.
when you say they were negative, there was no DNA typing produced as a result of putting them through the whole process?
That's correct.
And the substrate controls, the same question with respect to the substrate controls. Did each and every one of the substrate controls produce no typing result in this case?
That's correct.
Okay, and we'll enumerate them at a later point. You've written 2 reports in this case, is that correct?
Actually, 3.
Or 3?
The first report was a very brief one in last August for the Griffin hearing.
Okay, and then actually 2 reports with substantive results in them?
Yes,
And the material you're going to testify about now is a combination of the results that are reflected in both of those reports?
yes.
Okay. How many different RFLP gels did you run within those 2 reports?
3.
Okay, and do you have those numbered in some way?
Yes, I do.
And what numbers were they?
The numbers that we give them are, it's a sequential number. It starts with the letters AM for analytical membrane, and in this case, the numbers are AM616, AM625, and AM626.
And we'll put some of those autorads up in just a moment, some of those films, but what samples were on AM616? I assume that's the earliest of the 3?
Yes, those would be the 3 reference samples and also the extract of the SOC bloodstain 42A. That's the LAPD Matheson cutout.
That's the cutout GBM 13 or 13A?
Yes, 13A.
And so you had Nicole Brown, reference sample?
Yes,
You had Ronald Goldman's reference sample?
yes,
And you had the defendant's reference sample?
yes,
And then you had DNA that you extracted from the little tube that was cut out from the SOC 13A.
yes,
Is that correct?
yes.
And you did not do substrate controls because this is an RFLP run.
That's correct.
Were you able to successfully compare the pattern in DOJ 42 or GBM 13A, the cutout from SOC 13, with any of the 3 known samples in this case?
Yes, I was.
How many different probative, in other words, where you were matching or trying to match samples, how many different probative autorads or probes did you use in producing these results?
11.
Okay. Now, we have spent some time, considerable time, going over this case, haven't we?
Yes, we have.
How long, would you say?
Well, I think you and I were meeting about once a week since around the start of the new year.
Okay. And we've reviewed all these autorads,
Yes, we have.
and are some clearer than others?
Yes, I would say some of them are clearer than others.
Okay, and we've selected at least a few to show initially, is that correct?
Yes,
The better ones.
I think so.
And at a later point, we'll show all of them to the jury?
Sure.
Okay, Your Honor, at this time, I'd request to have Mark as People's next in order should be People's 269, and if my count is correct, it's A through I.
All right, 269A through I, are these autorads?
Yes, Your Honor.
All right, proceed.
I need to correlate numbers because we can't write on here, Your Honor.
Do you need some Post-its or something similar?
There are numbers on here. If we
Right.
can, I'll correlate the numbers on here with the exhibit numbers. Okay, we got A through 9. Mr. Sims, can you check, I want to make sure we have these grouped correctly, which autorads by the A numbers which you've labeled on relate to AM616? I've got A1 through 9 and A20 and 21.
I believe it's easier for me just to look at them because I don't
Sure.
have that separate listing.
I'll hand you what's labeled A1 through 9.
This entire stack is AM616.
Okay, and we've skipped A20 because that's not a probative one in this case.
I'd have to check A20.
That's
Those
that's the
numbers.
monomorphic probe.
Wait,
Okay.
wait. Excuse me, Mr. Sims and Mr. Harmon both have the habit of talking at the same time. Let Mr. Harmon finish asking his question before you start to answer. And Mr. Harmon, let him finish answering before you start asking the next question. Please.
Thank you, Your Honor. Now, does that represent the complete set of probative autorads for this case? For AM616, I'm
Why don't
sorry.
we just put the autorads for the sock up? Let's just do it.
Proceed.
I believe this is the entire set for 616.
And just to correlate them, Your Honor, 269A would be A1, B would be A2, C would be A3, D would be A4, E would be A5, F would be A6, G would be A7, H would be A8, and I would be A9. Are you with me, Mr. Sims? We're going A1 through A9. Okay. Can we, Mr. Sims, we're gonna start with a couple of the ones that we selected that have better clarity. If we would pick,
To strike.
I'm sorry.
His leading
All right.
and characterization. to
Sustained.
see
Raise
the
question. Jury should disregard the characterization.
If you would you put up one that's labeled A3, 269C? Or we'll give that to Jonathan to put up on the Elmo if we could, Your Honor?
Yes, Mr. Sims, you can step down. All right, this is 269C.
Okay, Mr. Sims, would you get us oriented on what we're seeing there? Describe those things that look like hash marks, if you would, and then we'll do the actual reference samples and the stain in a minute, okay?
Okay. This is an autoradiogram. It's an X-ray film that has the DNA patterns for this particular probing developed on it. What Mr. Harmon described as hash marks, we also call those ladders sometimes. You see that in those 4 lanes. And those are the molecular size standards. In other words, those are the knowns. Those are like the molecular ruler that you're placing on this gel along with the samples so that you can make measurements of the sizes of the bands in the lanes, in the sample lanes.
Okay, could you, from now on, those are very clear, but could you use the point maker when you're going to point out other things?
Okay.
Okay, let's start in the left-hand lane, inside that left-hand ladder, if you would, and just point to that and tell us what that sample is.
This particular lane, which has 2 bands, this is the upper band, and then there's the lower band. That is the K562 that I spoke of earlier. That's a standard that's used nationwide in laboratories, and we know what the particular sizes of that particular sample are so that we can use that as a quality control measure.
Okay, and the next lane to the right of that single band, what is that?
There's actually 2 bands here, and one of them is weaker on this particular copy than it is on the original. But this is the quality control sample. You can see a band for it here, and then there's a weaker band down in this particular region that appears on this autorad. That's pretty subtle on this overhead, but there is a band in that particular area, and that particular lane represents the quality control sample that I mentioned earlier. In other words, I don't know what the right results are for that particular sample. It went through the entire extraction process. I run it out on this gel. I make sizings of it, and then I submit those results in a blind fashion.
Okay, and that's the QC sample that the supervisor peeks at the list to make sure you got it right?
That's correct, and I believe in this case it was QC 806, but I don't recall exactly the number.
Okay. Now jump over, there's one ladder, and then there are the 3 reference samples, is that correct?
That's correct. The 3 reference samples are in a row. I'll just, I guess, point out where the bands are. The first sample is from Nicole Brown's reference bloodstain extract. The next one is from Ronald Goldman's reference bloodstain extract. and then the next one is from Mr. Simpson, the defendant's reference bloodstain extract. So those are the three reference samples that are on this gel.
Now, is this fairly typical when you have three unrelated people to have three patterns so different from one another?
This is
What-
very,
Hold on. What's the objection?
Objection in terms of relevancy and foundation as to what's fairly typical.
Call roll.
it is very typical because these probes have what is called a high degree of discriminating power, and people tend to be different. For example, in this class that I'm taking in my Berkeley program, we took, I think, 10
We've answered the question.
Okay,
Next question.
Have you recently discussed this in your class in Berkeley?
yes.
Hearsay, sustained. Let's move on.
Okay?
Okay,
Okay, so all three of those samples visually to you are clearly different than one another.
yes, they are.
Is that true?
Yes, they are.
And Robin Cotton described the first step in the evaluation process as a visual examination. Visually, when you saw this autorad, what opinion did you reach with respect to the consistency between Nicole Brown and the sock stain 13A?
I reached the conclusion that there was a visual match.
And let's just talk specifics about this autorad, and we could generalize with the next ones. Do you also do computer sizing in your laboratory?
Yes, we do.
And based on the computer sizing in this case, did your computer, and do you agree that those two samples are consistent from having come from the same source?
Yes, they are.
The two samples are Nicole Brown and the SOC 13A.
Yes,
And they're inconsistent with having come from Ronald Goldman, is that true?
that's correct.
And inconsistent with having come from the defendant,
That's correct.
is that true?
Yes,
And then on the right-hand side, there's another ladder.
that's correct on the outer flank there.
Now, this is from AM 616, and I believe you said there are 9 separate autorads that compare these samples. Is that right?
Yes, for this particular gel.
Okay, could we take a look at A5 now? I'm sorry, could we, would you just put arrows by the SOC 13A, the two-banded pattern, and the pattern of Nicole Brown? And may we capture that, Your Honor, and move on?
Let me redo that.
Not that.
Whoops.
Okay, you've actually identified 2 bands. Are those each two-banded patterns?
Yes,
And which probe is that from?
I would have to look at the actual autorad to know it, because I don't have all the patterns
Okay,
memorized on the
can we capture
K562.
that first and then have them, may that be,
That looks like D2S44. But it's all labeled. I mean, as I do these, I
Right.
label them as they come off, so it's at the bottom of the autorad.
okay, can we take a look at the autorad for D4S139, which is labeled A5? Would you put it screen, please? May we mark the print of autorad A3 for D2S44 269C1?
Yes. All right, Mr. Harmon, which autorad is this?
This is the autorad labeled A5, which represents the probe D5S110.
And this is in your sequence 269E.
This is 269E.
Thank you.
Now, Mr. Sims, this is, Robin Cotton explained how a membrane is probed and then stripped off, another film is put on, This is another film representing the DNA for this probe that comes from the same membranes. Is that true?
I don't mind getting through this, but maybe the witness can testify.
Yes. Pleading.
Sure. Are all of the samples in the same relative positions on this autorad as they were on 269C?
Yes, they are. This is just hitting it with another probe.
Okay, hitting the same membrane with another probe.
That's correct. You strip off the old probe, then you hit it with a new probe and you develop a new film.
Okay. And so the ladders are in the same relative position,
Yes,
and inside the left-hand ladder, that's K562,
yes,
and to the right of that, is that the QC?
yes,
And then once again, in the middle, the same three reference samples are in the same relative positions.
yes.
Now, can you simply look at that visually and determine that these three unrelated people, Ms. Brown, Mr. Goldman, and Mr. Simpson, are dramatically different than one another at this probe?
Yes, I think they are. I think it's clear.
And visually, were you able to determine whether or not Ms. Brown could be the source of the stain on the socket 13A?
Yes, I declared that a visual match.
And that Mr. Goldman was not the source of that blood?
Yes, I concluded that.
And Mr. Simpson was not the source of that blood?
Yes,
Could you mark arrows on the Nicole Brown pattern that you determined is consistent with the sock stain 13A? And also the sock stain? And did you follow up your visual examination with a computer sizing of those stains?
yes, I did.
And did the computer sizing determine that those two samples appeared to match one another?
Yes, it did.
May we capture that at
E1,
E1, Your Honor?
269 E1.
269 E1. Next, may we look at the probe labeled A6, which is for the probe D4S139 Each of these probes is a different marker, is that right?
That's correct. We're looking at a different area of the DNA.
And 269F, the autorad labeled A6, that's another film from the same membrane as the previous two films were from, is that right?
Yes,
And the ladders are in the same position?
yes,
The K562 is in that first lane inside there?
yes. It would actually be lane number 2.
Sure. And the QC is to the right of that?
Yes,
And there's another ladder?
yes,
And then, once again, the three reference samples in this case are in the same relative positions?
yes,
And visually, can you distinguish Nicole Brown from Ronald Goldman from the defendant in this case?
yes,
And were you able to make a visual determination whether Nicole Brown could be the source of the stain on the sock 13A?
yes, I did make that determination.
And were you able to exclude Ronald Goldman as the source of that stain?
Yes,
And were you able to exclude the defendant as the source of that stain?
yes,
Could you put arrows on the Nicole Brown pattern that you feel matches the sock on the stain on the sock? May we capture that, Your Honor, and may that be 269F1?
F1.
Mr. Sims, could you move the cursor arrow up to the front? Thank you.
Now, Mr. Sims, for purposes of illustrating this to the jury, those labels up on the top, those are not actually labels that you produce as part of the laboratory testing, is that true?
that's correct. Those are for court display.
Excuse me?
Those are for court display at the top.
And is that just another transparency that's put over them?
Yes,
Do you actually make records of what sample is in what is in which lane?
yes,
And is that labeling, is that actually a piece of transparency that's put over the autorad?
the labeling at the top?
Yes.
Yes,
It's a full sheet of it.
yes,
And I believe you've already mentioned there are 6 other different RFLP probes or genetic markers for this membrane. Is that correct?
yes.
And we will show them on the light box so the jury can see them with the court's permission probably tomorrow. But to summarize your conclusions or your findings with respect to those other 6 RFLP probes or genetic markers,
Could the witness, and you ask him a question instead of...
Actually, once you have, are you done with him down here?
I'm just going to finish this and start with the next series, Your
All
Honor.
right.
Did
Proceed.
you I'll withdraw my question, Your Honor.
Proceed.
Did you reach a similar conclusion with the next 6 RFLP probes or genetic markers, similar to the one that you reached with the 3 that you've demonstrated so far?
Yes,
And the conclusion was what with respect to those 2 stains?
that Nicole Brown could be the source of that sock on 13A.
The stain on the sock, 13A?
Yes,
Okay. And I believe you mentioned that the next membrane is labeled AM625.
yes,
Is that correct?
yes.
And how many autorads are represented, or how many autorads did you produce with respect to that membrane?
I'd have to check my notes on
Would
that.
you do that? Go, you can go on.
There were 8 probes on AM625.
Okay, and would you please list those probes for us? Or let me ask you a foundational question. We're using the term probes and we keep using these letters D. Can you explain what the D and the numbers and letters following that mean?
Yes, actually, when we're using these terms D, that describes what is called a locus, and that is the location of where this particular segment is found on the various chromosomes. The probes have names like YNH24, MS1, that sort of thing that usually describe who found them and what order they found them. It's a more personalized thing. The formal locus is the D, that stands for DNA. We talked about D4S139. 2 means it's on chromosome number 2. S means it's a single locus probe, so in other words, there's only one point in all the genome where this would light up, where the bands would show that variation. And then finally, the 44 is just a number on which it's registered. It's a sequential type of number for the mapping of chromosomes.
You mentioned these things are discovered. Are the discovery and description of these things published in the scientific literature?
Yes, this is a process that goes on before the forensic people even get to them.
And how and why does a lab such as yours select the different probes to use in casework?
The basis for the selection is that we want probes that are of good strength, good quality, good purity, but most important, we want them that show this tremendous amount of variation. In other words, we're looking at probes that have high degrees of discrimination, and that is that they are capable of distinguishing 2 individuals. And in fact, most of these probes have the capability of excluding about 95% of 2 individuals. 95% of the time, 2 individuals would be shown to be different by these probes.
No foundations for this witness.
Sustained. Answer is stricken, jurors to disregard. No foundation for that.
Is this something that you keep abreast of in the scientific literature, reading about the probes that you use in your casework?
Yes,
For example, are you familiar with the specifics or the qualities or characteristics of the probes that you've described so far?
Yes.
And if you could generalize, what are the characteristics in terms of differences between and among people?
I think that this is foundational in an area of population genetics beyond this.
Stan? Need some additional foundation.
How many articles do you think you've read that describe the different characteristics of the probes that are used in this case?
Several. I don't know the exact
And-
number.
And from reviewing the scientific literature, are you familiar with the characteristics of the different probes that are used in forensic work?
Yes.
Without being specific to the probes that you use in the case, what characteristics are desirable for use in forensics?
The desirable characteristic is one that is capable of distinguishing among individuals. In other words, a good probe would be a probe that can show a great deal of variation, large differences in band patterns across the population. So that means that two people randomly selected would tend to be distinguished by this particular test.
Objection as to, now he's
Over-
testifying.
Excuse me, it's objection foundation.
My apologies.
Overruled.
Foundation.
And based on your review of the literature and your familiarity with the probes that are used in these cases, is it also desirable that most of these probes produce two-banded patterns?
Yes, it is. That's called the degree of heterozygosity, how often you see two bands.
Um, Your Honor, at this point, I'd like to have marked as, or I guess we never got to you listing the probes that are on membrane AM625.
On the, yes.
Would you do that by A number as well as the D
Again,
number? Or
I would need to look at the A number because those relate to discovery numbers. Can I come down?
Sure. On AM625.
On AM 625, the loci that were examined were D1S7, D2S44, D4S139, D10S28, D17S79, D7S467, D5S110, and D17S26.
Okay, and was each of these probes selected because,
All right, does Mr. Simpson need to be there because his
No,
voice
I'm
voice
sorry.
doesn't carry well with his back turned?
but were these probes selected because they identify a spot on chromosomes where people differ from one another?
Yes,
The way you've described the literature recommends?
yes.
Overruled.
May the next group of probes or autorads, specifically A10, A12, and A13, be marked as 270, A, B, and C?
Yes.
which would be
270.
270A. Thank you, Your Honor. Now, what samples were on this gel that produced this autorad?
This particular autorad, or this particular gel, AM625, again has the three reference samples on it, Brown, Goldman, and Simpson. Then it has, it's a repeat analysis, or not a repeat, but an additional analysis of the same sock blood stain, 13A. And then finally it's got a sample on there from the glove that we talked about earlier, and that in particular that was stain G3, and that was from the inside surface back of the ring finger.
Okay, we'll show a board probably tomorrow that shows the exact locations of that, but where was that stain, G3?
It was on the inside surface of the glove on the back of the ring finger.
When you say inside surface, you mean the inside out?
That lining, yes.
Lining. And so could you hold up your hand and show the jury as if you were pointing at it from the outside? Okay, you've got your right hand up, the ring finger between the first and second knuckle?
Yes,
Okay,
somewhere up in that area.
okay. Now let's go left to right again and describe what's in the respective lanes. If you would, on the left-hand side is what?
On the very far left, we start with one of these size markers again, that's the ladder. Then lane number 2 is a K562 sample. Then lane number 3 is another quality control sample, another QC sample. Then we get into another, in lane 4 we see another one of the ladder.
Could you come back over here, Mr.
Sure.
Sims?
Sure.
Okay, just, if you could, just point to the ladder first on the left-hand side, okay. That left ladder, then the two-banded pattern.
And this pattern right here, this lane number 2 is from the K562. Then in this lane, we see another quality control sample.
Okay,
Then another one of these size marker ladders.
okay, now in the middle again, you have the 3 reference samples, is
Yes,
that right?
we have Brown, Goldman, and Simpson in those 3 lanes.
These are the same reference samples that were on the first gel for AM616, is that right?
Yes,
Okay. And once again, visually, can you distinguish Ms. Brown from Mr. Goldman from the defendant?
yes, I can on this autorad.
You don't need the computer to do that.
No,
Okay. Now, to the right of the 3 reference stains or the reference sources in this case is what?
well, there's one more size marker lane.
Okay.
And then in the next lane is the sock stain again, the same stain that was on AM616, the same extract, in fact.
Now, okay, the same extract, that's from the same cutting that Greg Matheson sent you?
Yes,
And it's part of the same extracted DNA?
yes,
And is this an example of what one could do if one retested extracted DNA?
yes,
That's what you did.
yes.
And let's focus on that, and if you would, I'm going to ask you to change colors when we get to the next stain. But visually, did you compare Nicole Brown with the stain on the sock, 13A?
Yes, I did a visual comparison of that sample there in lane 13A with Nicole Brown.
And did you determine that the bands matched?
Yes, they visually match.
And once again, Mr. Goldman was not the source of that stain?
Yes, that's
And-
true.
And the defendant was not the source of that stain?
Yes, that's
Is that
true.
Could you put arrows that show the comparison between Nicole Brown and the stain on the sock?
Mr. Harmon, do you want to consider maybe putting the arrows the other direction and on the other side of the lane?
Or are we going to fit all this in, Mr. Sims?
I
I think
think,
you turn the arrows the other way and put them on the other side of the lane.
On the Nicole Brown lane? Yeah, could you try that?
okay, you,
Okay, and did you follow up your visual examination with the computer sizing of those stain, of the stain and the reference sample?
yes,
Did you determine that those two samples appeared to match?
yes,
Okay, now, you have the stain on the ring finger from, that you've identified as glove G3. Visually, were you able to determine that that stain appeared to match one of the reference samples?
yes, it does.
Now, which one is that?
That would be the sample from Goldman,
From Ronald Goldman?
yes,
Okay, and did you follow that up with a computer sizing?
yes, I did,
And did that confirm that those two samples appeared to match?
yes, it did.
Could you change colors and put arrows that show the corresponding bands between Ronald Goldman and the glove G3?
Do you want the arrows to go the opposite?
It, whichever fits. Okay. And you put some pink arrows and those reflect the correspondence between Ronald Goldman's pattern for that marker and the pattern from the glove stain, is that correct?
Yes,
Um, I'm sorry, and that's the glove number 9, that's the only glove you got?
yes, that right-hand glove was
You
number
never
9.
got the glove from Bundy, number 37.
That's correct, we never received that other glove.
Could we capture that, Your
Yes.
Honor? And
And gentlemen, you're both talking over the ends of each other again. All right.
could we put on the projector the autorad A12 for the probe D4S139,
Yes. That'll be 270B.
270B, Your Honor? Okay, everything's in the same relative position, Mr. Sims?
Yes,
Okay, and once again, you have the K562 sample
yes,
inside the left ladder.
yes.
To the right of that is what?
And to the right of that then would be the quality control sample.
And to the right of that is what?
A size marker,
Okay, and then again, the 3 reference samples
yes,
in the same relative positions.
yes,
And visually, can you distinguish Nicole Brown from Ronald Goldman from the defendant in this case, based on these results from D4S139?
yes.
You don't need the computer to do that?
No, the eyeball is sufficient.
And once again, to the right of the 3 reference stains is what?
That again is the sock extract stain.
Well, first there's a ladder and then the sock?
Yes, I'm sorry, the ladder and then the sock.
And that's the same stain, that's from the same stain that Greg Matheson cut out and sent you?
Yes,
And were you able to visually determine whether or not that stain matched any of the 3 reference samples?
yes.
What did you determine visually?
I determined visually that it matched the sample from Nicole Brown.
Could you put arrows that depict that, please?
Yes,
I would suggest we reposition those last 2 since it appears the arrows are obscuring the actual band.
And did you follow up the visual comparison with the computer sizing?
Yes,
Did that, again, determine that Ms. Brown's reference blood seemed to match the stain from the SOC 13A?
yes,
And again, did you compare the stain from the glove number 9 from Rockingham, the stain designated by you G3, with the reference samples?
yes, I did.
And were you able to compare, determine visually whether any of the 3 reference samples could be the source of the stain at glove G3?
Yes.
Glove number 9. Which
Yes.
sample?
That glove sample matched the pattern for Ronald Goldman.
Could you put arrows to that effect?
Can I come at it at an angle?
Sure, we can try top to bottom here. Okay, now you've got some pink arrows on a diagonal.
Yes, those are for the Goldman sample.
And did you follow up that visual comparison with the computer sizing?
Yes, I did.
And once again, did you determine that Ronald Goldman reference sample appeared to match the stain from glove number 9 labeled G3?
Yes,
Did you capture that?
Yes.
May that be 270B1, Your Honor?
Yes.
And may we have A13, the autorad A13, which is for the probe D7S467. It's been labeled 270C. Okay, now these lanes are all in the same relative positions, the samples are.
yes,
Why don't you go through them from left to right?
again, lane number 1 is the size marker lane. Lane number 2 is the K562 standard. Lane number 3 is the quality control sample. Lane number 4 is the size marker lane. Then the next lane is the Brown sample. The next one is the Goldman reference sample. The next one is the Simpson reference sample. Then the next one is another size marker. Then we have the SOC 13A stain again, the same stain we've been talking about. Then the size marker, then the glove G3, and then finally another size marker.
And let's go back to the 3 reference samples. Again, is it easy for you to distinguish visually from among the 3 reference samples that those are from 3 different people?
Yes,
You don't need a computer to do that.
no,
And to move over to the stain from the SOC 13A, visually, were you able to determine whether one of the reference samples matched or appeared to match the stain?
yes.
Which one?
That would be the reference sample from Nicole Brown.
And could you put arrows on the bands from the stain SOC, or the SOC stain 13A and Nicole Brown's pattern? Okay, and similarly, did you compare the stain from glove number 9 from Rockingham that you designated G3 with the 3 reference samples?
Yes, I did.
And visually, did you determine that one of those reference samples matched the stain from the glove?
Yes, I did.
Which one?
That would be the sample from Ronald Goldman.
Did you follow that up with a computer sizing?
Yes, I did.
Did that continue to demonstrate that those 2 samples appeared to match?
Yes, it did.
Could you put arrows on that, please? May we capture that, Your Honor, and mark that 270C1?
C1. Yes.
Capture. Okay. Don't sit down, Mr. Sims. I just have a general question. You have the sock stain on both of these autorads, is that correct?
Yes,
So you ran part of the same DNA on two separate RFLP runs.
yes,
Now, initially you mentioned that there is a match between Nicole Brown and the sock stain 13A with 11 probes, is that what you said?
yes.
Okay, now the first series just showed 9 probes. Can you explain if there were 9 on the first series, AM616, and then I believe 8 in the second series, where the additional 2 probes came from?
Well, there is some overlap of some of the probes, and then there are 2 additional probes on 625.
Okay, so you had 9 on the first membrane,
Yes,
616, and some overlap and 2 additional ones on the second run.
that's correct.
Okay, in the third membrane, I believe you described it as AM626.
Yes,
What samples were placed on that membrane to run? Or on that gel, rather?
yes, those samples included 3 additional stains from the glove, as well as the reference samples from Nicole Brown and also Ronald Goldman.
Could you designate or give us the number that you assigned to those stains? And if you would, just turn to the jury and show us where on your hand the respective stains came from.
Yes, I can do.
Point that out.
These stains are designated G1, G2, and G4. Keep in mind, G3 was the one we just saw. So this is G1, G2, and G4 now. This is all in the glove. G1 is the inside surface of the glove. Again, we're working with the glove turned inside out. The back of the index finger, back of the index finger, that's G1. G2 is the side of the middle finger. And then G4 is on the back of the hand. It's in this general area in the back of the hand.
On the inside or the outside?
That's, again, on the inside surface,
So when we say inside, we mean the liner.
the liner.
Okay. And in the positions you pointed out.
Yes, and again, I believe there's a poster that will actually show these a little more clearly on the glove itself.
Okay. Mr. Sims, if you would, when you answer my questions, try not to look at me and address them to the jury. At this point, I'd like to have marked as
271.
271. Right now, simply 3 autorads from AM626. They're labeled A16, A17, and A25. And A16 represents the probe D4S139 A17 represents the probe D2S44, and A25 represents the probe D5S110. Can we have A16 up, please? And if you could get us oriented. If you start from the left and use the point maker, Mr. Sims.
Yes, this particular gel, AM626, represents these additional glove stains compared to Brown and Goldman's references. The other point I wanted to make about this particular gel is we're using less DNA than on the earlier gels. So this is now about 100 nanograms, something in that neighborhood, that was applied for these samples.
Is that why the bands in the right-hand columns that appear to be the evidence stains appear to be fainter?
That's one particular reason, yes. The other point is that these involve actual mixture samples. And as I go through these, perhaps we can talk about the different mixtures.
Okay,
That
but
we see.
starting on the left-hand side, we have the ladder.
Yes, starting on the left-hand side, lane number 1 again is the ladder. Lane number 2 is the K562. Lane number 3 is the quality control sample. Lane number 4 is the ladder again, the size standard. Then we have the Nicole Brown reference sample, then the Ronald Goldman reference sample, then another size ladder. And now we have the 3 stains from the glove, G1, then G2, then G4, and then finally another size marker.
Now, why didn't you have the defendant's reference sample on this gel?
Because he had already been excluded using the PCR tests.
And we'll talk about that in a while, but any test that excludes somebody at one marker, absent some other explanation, is excluded for other markers?
Yes.
Now, looking at the 2 reference samples between Nicole Brown and Ronald Goldman, can you easily distinguish between the 2 people?
Yes.
You don't need a computer to do that?
No.
Now, moving over to the glove, number 9 from Rockingham, stain labeled by you G1.
Yes.
How many bands are in that pattern?
There are 4 bands in that G1 pattern.
Could you identify those bands, or just point to them? We're going to label them in a minute, so let's not label them. If you just point out
1,
from top to bottom.
1, 2, 3, 4.
Okay, now, Dr. Cotton mentioned that we inherit one of these bands from our mother and one of our bands from our father. Is that correct?
Yes.
What's the significance of seeing more than 2 bands in a pattern like this?
Well, if you see 4 bands, that indicates that it's from more than one individual.
And
It's a mixture of samples.
It indicates that it's a mixture
Yes.
because there are more than 2 bands.
Yes.
Okay, and if you would, were you able to compare Ronald Goldman and Nicole Brown with the 4-banded mixture stain, and it's labeled G1 from the glove number 9?
Yes.
And what were the results of your comparison?
The visual comparison was that those 2 combined could produce the band pattern seen in G1. In other words, G1 could be a mixture of those 2 individuals' samples.
If you would, hopefully it'll fit, could you identify the pairs of bands that match up between, let's start with Nicole Brown, between G1 and Nicole Brown? Okay, and what can you say about the remaining 2 bands in that 4-banded pattern when you compare them with Ronald Goldman?
Those remaining 2 bands are consistent with Goldman's pattern.
Are you going to be able to fit that in? Could you change, let's change colors if you would.
Excuse me, I might want to come at it from an angle,
From the bottom?
maybe from the diagonal.
Okay. Now, is there anything about the nature of that 4-banded pattern in the lane G1 or in the stain G1 alone that helps you sort out whether or not those 2 reference samples in fact match or came from or the stain came from a mixture of those 2, the 2 reference samples?
No, in that lane by itself, it would be very difficult to determine what went with what,
Why is that?
because they're relatively equal in intensity.
And if the intensities were different, what would that help you do?
If the intensities were different, you could begin to see, you might see that one pattern tended to go with one individual, whereas the other two bands tended to go with the second individual.
But as it stands now, because there are no intensity differences between or among any of the four bands in G1, you're unable to say anything simply on that basis.
Yes, I think if you look at that G1 lane by itself, it'd be very difficult to say too much.
Do you want me to keep going?
No, let's be a good spot. All right, ladies and gentlemen, we're going to take our recess for the afternoon. Please remember all of my admonitions to you. Do not discuss this case amongst yourselves. Do not form any opinions about the case. Don't conduct any deliberations till the matter has been submitted to you. Do not allow anybody to communicate with you with regard to the case. And we'll stand in recess as far as the jury is concerned until tomorrow morning at 9 o'clock. All right, thank you.